Thanks, Timo. Yeah, as Timo said, I'm going to convince you that hypothermia is the optimal treatment to overcome tumor hypoxia. Before I start, I'd like to acknowledge the scientific collaborators that we've had on this project, the technicians from our group that probably did most of the work that I'm going to present, and financial support that we've got. Now, as you all know, tumor growth of solid tumors is limited. Tumors are like normal cells. They need a supply of oxygen nutrients. They get that from the host vasculature when they originate, but when they get beyond a certain size, they become starved of oxygen nutrients. And further growth can only happen if they develop a vasculature from the host blood vessels by this process of angiogenesis. And when that's established, you get uncontrolled growth of the primary tumor, and it's a major source of metastatic spread. Problem is that the vessels that are formed are very different from what you see in the normal tissues. This is a vascular casting technique that Peter Valpel borrowed from Maurice Kernodin, showing vessels in colon, subcutaneous, or skeletal muscle. And these are tumors growing in the same sites, and you can see straight away that they're very different. There's a load of differences, structural and functional abnormalities. Basically, it's a very chaotic, very primitive system. It doesn't have all the regulatory controls. It's just designed to supply oxygen and nutrients to the tumor cells. The other problem is that it doesn't develop as quickly as the tumor cells grow. So the tumor cells tend to outgrow the vasculature that it needs. And so you end up with this situation here, where this is a functional tumor vessel. Tumor cells grow around it in a corded structure, and then oxygen that the cells need diffuses out from the vessel. But as it diffuses out, it gets used up. So on the edge of this cord, you have these hypoxic cells, and we call them diffusion limited because they depend on oxygen diffusion, and they're chronically hypoxic because they stay hypoxic for a long time. We also know that flow through the vessels can fluctuate. These are measurements made over a 60-minute period in different regions. And if you get a shutdown in flow, in perfusion, all the cells become hypoxic, and we refer to this as perfusion-limited acute hypoxia. And that's basically our working idea for hypoxia. Now, the significance of hypoxia is illustrated very nicely in studies that use the oxygen electrode. It's an electrode that's inserted into the tumor. It's programmed, goes through in a stepwise fashion, making oxygen measurements, and you plot the relative frequency of the different oxygen tensions. And in normal tissues, you get this nice, normally distributed histogram with no low values. But in tumors, you can see it shifted to the left, you get hypoxia. And if you make measurements in head and neck cancer patients, prior to them getting radiation therapy, you can actually separate patients in terms of overall survival, whether they were less hypoxic or more hypoxic. And we know that oxygen is important for radiation response, so hypoxia is a radiation-resistant factor. Similar results were found in cervix, where measurements were made prior to them actually getting surgery. So now the hypoxia doesn't influence the treatment response, but what it's influences is malignant progression. The more hypoxia you have, the more aggressive the tumor is, and the more likely you are to see metastasis. So over the last 50 years, there's been a whole range of approaches for dealing with hypoxia, and you can divide them into different categories. You can increase oxygen delivery, and there's a number of ways to do that. You can actually sensitize the hypoxic cells to radiation. You can preferentially kill them. You can use agents that target the tumor vasculature, which can both increase oxygen delivery or decrease it, and as a result, kill the hypoxic cells. Or you can modify the radiation. You can do dose painting. If you can identify the hypoxic region, you can up the radiation dose to that region, or you can use high LET radiation where hypoxia is not such an issue. What you see is that hyperthermia actually crops up in most of these, except the last one down here. So what actually goes on? Well, this is some data. It's a combination of some old data and some new data we've collected. We've looked at tumor control in a C3H mammary carcinoma where we expose them to different doses of radiation. We take groups of mice, irradiate the tumor, put the animals back in their cage, and then observe them for 3 months to see whether the tumors are controlled or not. Low doses doesn't have any effect on tumor control. A high dose will give 100% tumor control. By varying the dose, you can produce a dose response curve. The dashed line is for radiation only. If you radiate and heat at the same time, you can push this curve way over to the left. If you introduce an interval between the radiation and heat, you only get a small difference. And you can calculate the TCD50 value, which is the tumor control dose to control 50% of the animals. So you just draw a line across here and then come down and read it off. And you can take the ratio between the response for radiation alone to the different heat treatments, and you plot this so-called thermal enhancement ratio. And this is varying the time between the treatments. So this here, 0, is a simultaneous heat and radiation treatment. So you heat for an hour, in the middle of the period, you just apply the radiation. So we're heating at this temperature. And you can see the thermal enhancement ratio for the tumor, so dark circles, is the best. When you introduce an interval between the heat and radiation, you can see the effect falls off, but it just plateaus out. It doesn't seem to matter if you give the heat before or after irradiating. But it never comes back to 1, so you're still seeing some effect. This is sensitization. This is effective cell killing. Interestingly, in normal tissues, and here we've got skin, you see the same degree of sensitization with simultaneous, but the drop-off is steeper. So you actually see no effect when you do this so-called sequential effect. And what's plotted here is the thermal enhancement ratios for a simultaneous heat and radiation or a sequential, where you've got a 4-hour interval. And you can see there's a clear time-temperature dependency. The longer you heat and the higher the temperature, the bigger the effect in both of these, although the effect here is less than what you see over here. So you see sensitization. How good is the sensitization by hypothermia? When you're giving, if I go back, when you're giving this simultaneous heat and radiation treatment. Well, we've done similar studies using classic radiation sensitizers. These are drugs that we designed and developed to target the hypoxic cells and sensitize them. This is one particular agent. We're doing tumor control again, radiation only. We're giving an agent called nimorozole, and you can see what happens. And again, you can calculate the enhancement ratios, and these are plotted here for 3 different sensitizers. Misonidazole, which is the classic lead compound back in the 70s, and that's shown by the open circles in this line. The more you gave, the bigger the effect. This is drug dose. Nimorozole is shown by the squares. It comes up and then sort of flattens out. It's not a good sensitizer as misonidazole, but it's less toxic. Misonidazole went into clinical trials but failed because you could only give doses down here before you ran into toxicity issues. Nimorozole is not so good a sensitizer, but you can actually give higher doses because it's non-toxic. Doronizole we've looked at, and that's this line here, has the same sensitizing ability as miso, but the same toxicity profile of nimorozole, and it's the new drug in development. But what I've plotted on here is the sensitization you see with heat. And you can see that this is the clinically relevant doses you see with these sensitizers. But you can see you can get that with 41.5 degrees heat. And if you can up the temperature, the effect gets even bigger. And see what you get with just 42.5, if the sensitization is massive. What's shown here is what happens if you combine hypothermia with misonidazole. So this is the enhancement ratio for misonidazole only. This is the effect of heat plus miso, 42.5 or 43.5. So you get an effect of the heat alone. It looks like it's just additive, which suggests that maybe the sensitization that's being produced by hypothermia is not necessarily the same as what you see with a classic sensitizer. And in fact, we tried to look at that over here by combining heat with nicotinamide. Now, what nicotinamide does is actually stops this happening. So it gets rid of acute hypoxia. We don't know how it stops it, but if you've got nicotinamide there, you don't see acute hypoxia. This is the radiation-only curve. If you give nicotinamide, you can see you enhance the radiation response. Enhancement ratio is about 1.22. If you heat 4 hours after giving radiation, you see an effect. 1.66 is the enhancement ratio. But when you put them together, the effect is even bigger, suggesting maybe an additive effect, which then sort of confirms the effect that this is what the sensitizers are working on, primarily, but hypothermia is probably working on the chronically hypoxic cells. How does it sensitize? Well, there are a number of suggestions. The most obvious, it improves tumor oxygenation. It can improve blood flow during the heating period. This is some of our own data where we looked at what happens in oxygenation status during the middle of a heating period at 41.5 degrees. You can see it goes up. When you turn it off, it comes back down. This is low values showing that they are reduced. There's some suggestions from Vujicic and songs suggesting that maybe the effect is maintained for longer. We don't seem to see it, although we went out to 4 hours, so it's a bit controversial. But they clearly show that during the heating period you see an improvement in sensitization. So that certainly goes on there. The other mechanism, of course, is that you see an effect on DNA repair. So this is an excellent review from 2001 that looked at it, looked at different assays for looking at repair, what mechanisms they looked at, and whether heat actually inhibited. And you can see that basically for all these except this one down here, you saw an effect. This is just a summary of the DNA repair mechanisms. So with radiation, you're getting single-strand breaks that can be converted into double-strand breaks. You actually see double-strand breaks from the radiation itself. And then these are repaired by different mechanisms. Two major ones, non-homologous end joining or homologous recombination. And it seems like hyperthermia, by affecting the proteins involved in this, can actually inhibit these repair processes. So that's sensitization. What about cytotoxicity? Well, that's been known for ages that heat is a cytotoxic agent. This is some work from Rosenthal and Piro looking at cell survival in vitro after heating at different temperatures for different times in human or mouse cell lines. And you can see a time-temperature relationship, and you get this sort of Arrhenius plot when you plot the slope of these values here with this breakpoint here. This is in vivo data. It's looking at time to reach 5 times treatment volume. It's actually, these numbers get bigger as you go down. It's plotted upside down. And the reason for that is they wanted to compare it to what you see in vitro. And again, you get this Arrhenius type plot that fits very nicely with that. So you see cell killing. Again, hypoxia seems to be a factor involved in that. This was some old data looking at what happens to cells exposed to 42.5 degrees for different times at normal pH and oxygenated conditions. But if you make the cells hypoxic, you see increased cell killing. It actually got bigger if you drop the pH. And this was some work from Gerwick and colleagues where they looked at the effect of pH, and it seems to be associated. So it seems to be more of a pH-dependent effect. But what you're going to find is that the pH, the acidic conditions, are actually going to be found in the areas where you have the chronic hypoxia. So it fits again with the idea that that's targeting those cells. You can actually look at that by modifying blood flow and see what happens. This is some work we did where we gave a physiological modifier that shuts down blood flow transiently and then recovers, or a vascular disrupting agent. If you heat tumors at 42.5 degrees for different times, you get a linear response in terms of time to reach 5 times treatment volume. The more damage you do, the longer it takes. But if you heat at the time of when you see the maximum shutdown with these agents, for hydralazine, you can see a change in the slope. With DMXAA, you get an effect on its own, but a change in the slope again. You can plot those slope values here for heat alone, heat with hydralazine, or heat with the VDA. Suggest the VDA might be better. If you take a temperature, say 41.5 degrees, it gives you an effect with the 41.5 degrees heat alone. It's similar to what you see with 42.5 for heat alone when you give the VDA or the vascular modifier. And we reckon that 20% of that effect is due to better tumor heating. The other 80% is changing the microenvironment and increasing the cytotoxicity. But there's another thing that goes on with hyperthermia, and that is that it induces vascular damage. This is some of our own data in a C3H mammary carcinoma, where we looked at perfusion during a 1-hour heating period at 42.5 degrees and after. And you can see this transient increase during heating, but immediately after, perfusion shuts down. Song and colleagues have shown similar data, although there seems to be a different temperature dependency. 42.5, they didn't see much. They saw vascular collapse at 44.5. So there's clear temperature dependent effects. But what you do is, if you have a functional vessel, you damage it, you shut down flow, and all the cells will die. And if you look at histology, in section as a function of time after heating at 42.5 degrees, you start to see edema first, which is indication that you've damaged the vessel, so fluid is leaking out. You'd start to see bleeding, and then a decrease in the viable tissue. And if you look at necrosis, that increases with time. And again, the cells that are most likely to die first are those that are already oxygen nutrient deprived, the hypoxic cells. So you have a direct effect of heat on killing cells and an indirect effect. The only place where you don't really see heat here is with high LET radiation. But you've already heard that maybe heat combined with low LET can be as good as high LET. This was the study that's been set up. Nino Data and Stefan Bodis have set it up where they're looking at hyperthermia with protons to see whether it gives the same as carbon-ion therapy. It's the Hypoxia protocol, hyperthermia, protons in sarcomas. This is the treatment schedule. And I won't go into details about that because there's a talk this afternoon that deals specifically with that. But it may be that even though that last if I go back, this last category, hyperthermia, isn't in here. It could actually play a role in that. This is a summary, just looking at the it's a meta-analysis we did of randomized clinical trials of radiation plus or minus hyperthermia for local tumor control, showing the odds ratio that you get. And of course, if the values are above 1, it's a significant benefit. And you can see for all these tumor types, you basically see the average, the odds value is above 1. The errors for this one overlap, so it's not significant, but it's a small number of patients. But when you put them all together, you see a highly significant benefit. I put this one in because it's something that I think is very interesting. It's from the Dutch group, where they compared over survival in cervix cancer when they can give radiation and heat compared to the classic standard treatment for cervix, which is radiation and cisplatin. It basically get the same effect. And the nice thing about that is you don't have the cytotoxic side effects that you get with cisplatin, and they can be quite horrendous. So you get the same tumor response and overall survival without those side effects. And I think that's something that tends to be forgotten a little bit. So this is the last slide. It's a summary of the conclusions. We have tumor hypoxia. It increases resistance to therapy and leads to a high degree of malignancy. There are traditional approaches for dealing with it: increasing oxygen delivery, sensitizing, bioreductive drugs, vascular targeting agents, modified radiation. And if you use those, you decrease hypoxia. And that will improve response to therapy and decrease the formation of metastases. But we can get the same effect with hyperthermia, either through hypoxic cell killing, direct cell killing, or indirect cell killing as a result of vascular damage, or radiosensitization by increasing oxygen delivery, inhibiting DNA repair, or combine it with low LET radiation. And that's the last slide, and I'll say thank you. You're all convinced it works, right? Speaking to the converted.
Yeah, so hypoxia, so hyperthermia and radiation are actually good partners then.
Yeah.
So one thing hyperthermia does is also at a reasonable temperature of 42, 41 is opening up the vasculature with respect to the lining. And so you get that you have more fluid going into the tissue.
Right.
So how does that affect the hypoxia status?
Yeah, it will do two things. One, I mean, you're going to get, with a low hypothermia, you're going to get initially, you're going to get an increase in flow. And that can improve oxygen delivery, of course, but it's a very short time.
Yeah, but you also get an increase
in... But then you're going to get, as the temperature goes up, I don't think you see it so much with 41.5. I think if you're going up to 42, 42.5, you start to induce some sort of damage to the endothelial cells. Once you start to do that, they start to roll up, they go into apoptosis, that opens up gaps, and then you're going to get edema. And when you're going to get fluid coming out, the interstitial fluid pressure in tumors is pretty high already. When that fluid comes out of the vessels, it's going to cause the vessels to collapse. That will induce hypoxia.
Okay.
Right?
Okay.
And that could be a complication. At the same time, the normal hypoxic cells that are already on the verge of dying, they will die because you've taken away chronically hypoxic cells. Chronic hypoxic cells are not really a problem. They're doomed to die. They're far enough away from the vessel. With time, they'll move away and they will die. They only become a problem when you start to irradiate and you can increase oxygen delivery and stuff. But you do have that issue that maybe there's a transient increase in hypoxia that could become a bit of a problem.
Okay.
Yep.
Are there other questions from the audience? Yes?
I read on one of your first slides a characteristic of tumors is an increased hematocrit. You have shown this.
Yes.
But on the other hand, we know that tumors are typically have a higher water content than normal tissue. So that hematocrit should be lower, I think.
No, but what you do is the flow through the tumor vessels can be slower, so you get this accumulation of red blood cells.
Oh, you mean the hematocrit
it's the hematocrit in the vessels, yeah.
In the vessels, okay.
In the vessels, yes.
Oh, that's okay.
Yeah. Do you want to use the microphone?
If you have
damage in your vessels.
Yeah.
I'm not that known, but has bleeding within tumor been described as a potential risk factor of hyperthermia? Because then you could expect that there is a bleeding issue as well.
Yeah, it's a good question. I mean, you are going to get bleeding. You get vascular shutdown. That's going to lead to a problem. It could be, I guess, if you're going to do vascular damage, you're going to see hemorrhagic necrosis. And in fact, if you do histological sections, you do see red blood cells in the regions of necrosis. So you've had bleeding in there, but it seems to be downstream of where the blockage is. So you're causing a blockage, it's downstream, you're getting bleeding into the tissues, but then those tissues are going to die as a result of it. So I don't think it's a problem. You would think that maybe if you're going to get bleeding, you could increase metastatic spread and stuff like that, I assume, but that's never been shown. So I don't think there's an issue there. Yeah.
Okay, thank you. If there are no